This video is about nerve tissue. We've already learned about epithelium. We'll also be learning about connective tissue and muscle tissue. The goals of the nerve lab are to recognize nerve cell bodies and their processes, which are axons and dendrites, and also to recognize the supporting cells that in general we call glia. We'd also like you to learn how to recognize the central nervous system as compared to the peripheral nervous system. And finally, within the peripheral nervous system, we'll cover how to distinguish epineurium versus perineurium versus endoneurium. The first slide we'll look at is a very low magnification light micrograph of the spinal cord. You can tell it's low magnification, as we learned about in the cell biology lab, because you can't see any cellular detail. The small dark spots that you can see in this more central portion of the spinal cord are not nuclei, they are in fact cells. So that's what makes this such a low magnification micrograph. The thing to note about the structure of the spinal cord is that the nerves are in the central portion, whereas the axons and other parts of the white matter are in the periphery. As you'll see in your neuro course, the reverse is true in the brain. Just to orient you, up here we have the dorsal portion of the spinal cord, and over here we have nerve rootlets for the sensory nerves that are coming into the dorsal portion of the spinal cord, and we'll look at dorsal root ganglia in a few slides. Here we have the ventral spinal cord. This is where the motor nerves are found. This is true bilaterally. There is a central canal filled with cerebrospinal fluid, and this is the ventral portion of the spinal cord, and some of the things you see down here are the ventral rootlets coming out from the motor neurons. Now we're at high magnification light micrograph, and what I'm sure you're noticing immediately are these very large cells with very large euchromatic nuclei and very prominent nucleoli. This is very similar to what you saw in the cell biology video. So these very large cells are the motor neurons of the ventral spinal cord that we just looked at. And these motor neurons are full of what we call Nissl bodies, and they're named after a person who first described them, and they represent ribosomes, both free polysomes and rough ER. And this particular section is stained with crystal violet, which highlights the ribosomes and the nucleic acids. But we don't see here cell membranes and other organelles. Outside of the large motor neurons, you'll note that there are 2 or 3 different cell types. One is a fairly prominent nucleus. You don't really see the cell processes here, just the nucleus. It has kind of an oval nucleus. It's euchromatic. These are nuclei of astrocytes, which is one of the types of glia. Compare that to a smaller, more heterochromatic, a bit more round nucleus. This is the nucleus of an oligodendrocyte, which myelinates axons in the central nervous system. Up here you'll see a third cell type, and that is the endothelial cell wrapping around this blood vessel, which even though we don't know the vessel types yet, this could be a capillary or venule, but you don't need to worry about that. Just be able to identify the endothelial cells that we learned about in the epithelium lab. So by comparison, this is the micrograph at high magnification of the peripheral nervous system. So let's step back just for a moment to the central nervous system. One of the things that is true of the central nervous system is that it has very little to no connective tissue in it. We haven't learned about connective tissue yet, and we will be discussing that in the next lab. But the central nervous system is set up such that it is protected at its periphery by a number of layers in a structure that is called the meninges, which you'll learn about in detail in gross anatomy. And it's the structure of the meninges and the structure of the astrocytes in what are called the subpial glia that protects the nervous system and essentially eliminates the need for connective tissue between the cells, which means that you can really pack in cells very tightly in the central nervous system. By contrast, in the peripheral nervous system, what we see are these very large neurons, and the cell body has a comparably large nucleus and a very prominent nucleolus. And the staining of the cytoplasm using this particular stain, which stains membranes as well as other extracellular structures, is full of the rough endoplasmic reticulum and polysomes called the Nissl substance. And what you will note is here in this one particular area, that's an area that is free of this Nissl substance. That is because this is the beginning of the axon. It's kind of a staging area for those molecules and organelles that will be sent down the axon. Now notice the amount of connective tissue that exists, indicated by the red staining outside of the cell. And there are also nuclei which actually almost indent upon the neuron cell bodies. These are called satellite cells, and they very carefully wrap around every single neuron cell body in the peripheral nervous system. So if we go back one and start at the axon hillock, which is right here, this is the beginning of the axon, and when each neuron has only one axon, but it can have many dendrites, and we'll come back to this later in the lab. As that axon leaves the neuron, it will often be myelinated. And the next micrograph, which is at low magnification, it's very difficult to see any sort of cellular detail here, but you see all of these darkly stained structures with pale centers, and they're cut in various planes of section. So over here, they're cut in transverse or cross section, whereas just to the right, they're cut in more longitudinal section, and in other places, they're cut sort of halfway in between and what we would call an oblique section. And the way that this particular micrograph is, or the section is made, is that you can see this is a very large nerve, such as the sciatic nerve in your thigh, in the back of your thigh. And as you go across the nerve in cross section, you see many, many myelinated axons, the myelinating, or the myelin being this dark staining substance. And the clear area in the middle is the cytoplasm of the axon, which is filled with neurofilaments and microtubules. You can't see that at this magnification, and don't worry about that, but we'll look at things a little bit later where you will be able to see those structures. Now note on the outside, there's an area that's pale stained. This is called the epineurium. And then there is an area which is more darkly stained and is cellular, which we'll look at a little bit more carefully under the electron micrograph. This is called the perineurium. And all of this pale blue substance in between the myelinated axons is endoneurium, or loose connective tissue. This is a similar section, but with a completely different staining method. So if we look here at the periphery of this nerve in cross-section, we see a dense connective tissue. And then there's this very darkly staining line, which in this low magnification light micrograph represents the perineurium, which we'll see later has 1 to 5 cell layers that completely isolate the axons on the inside from any peripheral substances. In the middle of this large nerve, you can see many, many myelinated axons in cross-section. Here is a blood vessel which is supplying the axon, and the dark staining in between is endoneurium. The preparation of this tissue has extracted a lot of the myelin. That's why it doesn't have the same appearance. Here you can see the myelin, and in this particular structure, which highlights the 3 layers of the three neuriums, if you will, the myelin has been extracted, but you can see the remaining structure of the individual axons. As the axons go out into the periphery, they run in the connective tissue space, which we'll learn about in the connective tissue lab. Within the connective tissue space, you will find blood vessels. So what I'm circling right here is a larger blood vessel, probably a venule, that contains blood cells, which happen to stain blue because of the staining methodology used. Here is another lumen, also a blood vessel. Here's another lumen, also a blood vessel. Here's another lumen down here, which because of its appearance might be a lymphatic vessel. It's kind of hard to tell. And if we are uncertain about what structures are, we're never going to test you on them. What I would like you to look at in this particular slide, in this longitudinal section of a peripheral nerve bundle, is that this is a structure which is surrounded by perineurium. So this is now a medium to high, or probably a medium or intermediate magnification light micrograph. You can certainly see nuclei. So here's a flattened nucleus, there's another flattened nucleus, and these are the nuclei of the perineurial cells. And inside the perineurium, you don't see a lumen, so that helps you recognize that this is not a blood vessel, and it is surrounded by cells, so it cannot be connective tissue, and because there is no lumen, it is not an epithelium. So by a process of elimination of the 4 different cell types, what we're seeing here is a peripheral nerve bundle, probably out in a place such as the hand or the forearm, and it's running in dense connective tissue. What are all of these individual nuclei that are inside the peripheral nerve bundle? These are the myelinating cells called Schwann cells that wrap plasma membranes around axons as they travel to their destination. Up here, we see a similar longitudinal section of another peripheral nerve bundle, again by comparison with blood vessels, by comparison with the surrounding connective tissue, by its lack of anything that distinguishes it as muscle. This structure can be determined to be a peripheral nerve bundle. At the terminus of the axon, or bundle of axons, you find a synapse. And this is a preparation where the nerve has been stained with India ink. It's very low magnification, and you can see that it is synapsing on a striated structure. In fact, this is skeletal muscle. And as the nerve branches, you can see these places where the nerve gets constricted. These are called the nodes of Ranvier. And as you will learn in detail from Dr. Robertson, the action potential jumps in a saltatory fashion from one node of Ranvier to the other. This allows very rapid conduction of the action potential. Also, please note that as the nerve contacts its target, in this case skeletal muscle, it branches extensively. And the principle here is that you want to maximize the surface area of contact between the nerve and the muscle cell. Now we're looking at a very high light micrograph image of the cortex of the brain. What we would like to have you look at here is a cell body, which I'm indicating circling right here in the middle. And note that it has a number of processes. There's 1, 2, at least 3, 4 different processes, maybe more. This is a rather thick specimen, and we don't see all of the details. And indeed, we don't see all of the cells. This is a rather unusual preparation in which, when a cell takes up the dye, it takes up the entire cytoplasm is filled with the dye, showing the tiniest little structures. And you may be able to see that as we go along this structure, which is called a dendrite, that there are a whole series of small bumps called dendritic spines. And these dendritic spines are increased surface area for synapses. So imagine, if you will, within the brain, that along the entire length of this apical dendrite and the other dendrites that exist on this neuron, that there are literally thousands of other axons synapsing, and that leads to integration of information in this cell body, then leading to an action potential which comes out into this single process called an axon at the base of the cell, and that axon then carries information to other neurons. Now we are looking at an electron micrograph, and it's a relatively low magnification electron micrograph because you can see entire cells, and here's a nucleus, there's a nucleus. And you notice this very thick, dark staining structure. This is myelin. These are the membranes of the Schwann cell. Here is the nucleus of the Schwann cell. Here is the cytoplasm of the Schwann cell. And here is myelin, which represents many, many layers of the Schwann cell plasma membrane. In the middle is the cytoplasm of the axon. So this is the cytoplasm of the nerve. And you'll note that within this cytoplasm of the axon, there are small organelles. These are mitochondria, which actually travel up and down the axon to the terminus and back to the cell body. Also, you should note this structure right here. Here is the nucleus of a Schwann cell, but you note that there is not myelin around the structures that it is wrapping. This is called an unmyelinated axon. And in fact, what is happening is that the Schwann cell is cradling 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, approximately 11 or 12 unmyelinated axons in little troughs in its plasma membrane as a way of protecting them. And all of the connective tissue in between is called endoneurium. So this is high enough magnification. We do not see the perineurium or the epineurium. All we're looking at are myelinated and unmyelinated axons in cross-section. Now we're at high magnification electron micrographs. You can only see parts of cells. Here is a myelinated axon. Here's the myelin. And in the middle, you see mitochondria. And in cross-section, you see small structures. These would be either microtubules if they have a hole in the middle, or they would be neurofilaments, which provide structure to the axon, and also the microtubules are the way that organelles and vesicles get transported up and down the axon. Just outside the myelinated axon is endoneurium, and then you have this cellular layer. This is a very good representation of perineurium. Here is a perineurial nucleus, and you can see that it's very flattened. You also can see that there is a cell here. You can't see the nucleus. So it looks like there's 1, 2, 3, maybe 4 layers of perineurium, and their job is to insulate what's inside the nerve from the outside world. And as you'll learn from Dr. Robertson in her talk about neurophysiology, this is absolutely essential because you have to control the ionic environment, which immediately surrounds the axons in order for them to work the way that they are supposed to. And of course, on the outside, we have a denser connective tissue, and this is what's called epineurium. So what I'd like you to do now is to go to the Nerve Lab and complete your study of all of the images. And once you've done that and have tested yourself using the unlabeled images, you can go to a nerve quiz, which is on Learn at UW, that will help test your knowledge of nerve tissue micrographs.